Hi, I'm Witkowski. I'm Director of the Banbury Centre here at Cold Spring Harbour Laboratory, and I welcome you to the 81st Annual Cold Spring Harbour Symposium on Quantitative Biology, meetings that have been going since 1933. This year's topic is targeting cancer, and I'm delighted to have Bob Weinberg from the Whitehead Institute with me on a rather overcast afternoon here at Cold Spring Harbour. So Bob, we were chatting a bit earlier that the first symposium where the word cancer-related appeared in the title was the 74 tumour virus meeting. And I remarked that to me that looked very much just to do with the biology of viruses rather than much to do with cancer. And you were at that meeting.
Yes, and at that time tumour viruses were studied in no small part because they apparently had an interesting way of copying themselves, and it was almost incidental that they also caused cancer.
Because that was only, what, 3 years after reverse transcriptase?
Yes, but again, people like me, and there were many others like me, looked at tumour viruses because they would clone genes for you in their own genomes. And it was just a sideshow that they happened also to generate tumours in animals. That was almost a distraction for a while.
Well, that changed significantly only 5 years later with the 1979 Symposium on viral oncogenes. What had led to this transformation in just that short 5-year period?
Well, there was the 1976 discovery in the Varmus Bishop Lab, University of California, San Francisco, of this astounding fact that a virus that caused sarcomas, connective tissue tumors in chickens, actually had acquired that ability by extracting and kidnapping and remodeling a gene that resided in the normal chicken genome, ostensibly because its role in the normal chicken genome was to help a bird be a bird, and therefore having nothing at all to do with the whole process of developing cancer.
But the idea, I think, it's very hard, even 40 years later, put oneself in the mindset of that period. But the idea that there could be in normal cells genes that, when mutated, could lead to cancer, and that viruses could transduce these, that was heretical, maybe too strong a word.
Well, it was way out there. It wasn't in the center of everyone's thinking. There was a consequence of that discovery, and the discovery that preceded it by several years, and that was the fact that it was becoming increasingly apparent that a virus which might have a small number of cancer-causing genes could, when entering into a cell, dominate the behavior of that infected cell, that cell ostensibly having tens of thousands of genes. So here, a single or a small number of viral genes could totally reconfigure the whole biology of an infected cell, converting a normal cell into a cancer cell. That itself, by that time, had begun to be accepted, but it was hardly obvious because the alternative, more intuitive hypothesis was that if you asked how many genes needed to be altered in a normal cell in order to make it into a cancer cell, it might have been dozens. And the fact that it was only a single one, at least in these viral models of virus-induced cancers, totally rechanged everybody's thinking, because suddenly one could understand the process of transformation, quite possibly, by studying a small number of master genes.
And there was another consequence of that paper, or an important part of it was that they showed that Sark had a common evolutionary, not evolutionary history, but the Sark gene could be found in the cells of different species. There was an evolutionary conservation.
Indeed, in the genomes of all animals, and as I recall at one point in the late 1970s, even in the genome of a sponge, which is, by all criteria, a very primitive animal. So this suggested, in a very compelling way, that the Sark gene was important in the biology of a wide spectrum of normal organisms, and that its involvement in cancer was only a rare aberration.
So let me read you a quotation from Peter Duesberg's. I guess it was he did a sort of summary of retroviruses, whereas Lionel Crawford did one on DNA tumor viruses. Anyway, Duesberg wrote, "A critical test for the viral transduction hypothesis would be to isolate a viral transforming gene from a normal cell. So that's a cue drum roll for your work. When you first isolated the human oncogene.
Yes, well, interestingly, the work I got involved with really wasn't that interested in tumor viruses. Much of my interest came, interestingly enough, I would say, from a study by Bruce Ames, who was then and is now at the University of California in Berkeley, who showed that if you looked at a whole series of chemical carcinogens, their potency in inducing normal cells to become cancer cells was actually reflected in the ability of these various chemical agents to damage DNA. And in fact, in retrospect, maybe the correlation was a little overblown, but at least in my mind and in the minds of many others, I assume, it suggested the inescapable conclusion that within the DNA of a cancer cell, there lay damaged, mutated genes that in some way were responsible for the abnormal behavior of that cell. That really was the idea that, or the lesson, the take-home lesson I took from Bruce Ames's work.
So again, with hindsight, it seems obvious that if there is a gene like the normal cellular equivalent of SARC in a cell, that could be undergo mutation that would activate it in a way that the viral oncogene did when the virus infected a cell. But the Bruce Ames thing, of a chemical damaging, in a sense, inducing mutation in the cellular gene, that had a particular potency for you?
Well, it did have a particular potency because retroviral carcinogenesis by already the mid-1970s was seeming increasingly implausible, virus-induced cancer, as being a major factor in creating human cancer. Right. On the contrary, chemically induced cancer, including, for example, the cancers induced by cigarette tobacco tar, that seemed increasingly to be a more plausible agent for causing human cancer. And so certainly by 1977-78, at least I became convinced that retroviruses were interesting, but they were not going to lead us directly to understanding how chemically induced human cancers, and I believe many of the spontaneously arising cancers one sees in humans, how they arose. I thought there might be a much different causal mechanism.
What strategy did you use to isolate, to clone RAS?
Well, it turned out we had been working on viruses, on retroviruses, of the sort that Howard Temin had first discovered, of the sort that Varmus and Bishop had first studied in great detail. And we acquired certain laboratory skills, including the ability experimentally to take DNA from one cell and put it into another cell. And the DNA that was moved from one site to the other actually entered into the second cell and began to function as if it were now an introduced gene, ultimately a native gene of the recipient cell. And so in 1978, I all of a sudden thought, if we can do that by taking DNA from a virus transformed cell and putting it into a second cell, which then became converted from a normal cell into a cancer cell in response, maybe we could do the same thing if we extracted DNA from chemically transformed cells, which had never experienced a viral infection. To the extent that there was a cancer-inducing gene there, it was with great likelihood a mutated version of a normal gene that somehow had undergone damage or corruption as a consequence of it having been exposed to, for example, a chemical carcinogen. So that was the thinking. It was way out there, not because it was so conceptually complex, but simply because experimentally it was quite challenging. And so starting in 1978, a fellow in my lab began the onerous task of extracting DNA from chemically transformed cells and introducing that DNA into previously normal cells. And it was experimentally challenging, but lo and behold, after many months of trial and error, he actually discovered that one could do that. And that ended up being, in my own career, by far the most important set of experiments, because it revealed a simple and obvious truth that in the DNA, in the genes of a chemically transformed cell, and by extension human cancer cells, in that DNA there must lie a single gene, a small number of genes, that were responsible for choreographing, for orchestrating the cancerous behavior of the cells in which they resided.
And of course, once one could start isolating, let's still call them oncogenes, people then started examining the proteins and
Yes.
the functioning. And coming back to the symposium, there was the 1991 symposium on cell cycle, where the effects of these genes on controlling cell division, cell cycle, became evident. And again, with, I guess it was in the 94 one that Ed Harlow thought that the elucidation of signaling and signal transduction pathways had become a critical aspect of studies of cancer, again arising from the ability to clone cancer genes and examine what their proteins were doing.
Underlying all that, of course, was the ability acquired first in the late 1970s to actually clone a gene, that is to say, to extract a single gene from amidst tens of thousands of genes with which it co-resided within a single cell, either a normal cell or a cancer cell. So by the 1990, one had acquired great facility in doing so, and it also acquired the ability to study the resulting proteins. Keep in mind that the earlier work I discussed before indicated that DNA could transform from a cancer cell, could transform a normal cell into a cancer cell, but yield no insight whatsoever into how, in terms of mechanism, this introduced cancer gene worked. And that is exactly what Ed Harlow was referring to in the early 1990s. That is the biochemistry, the protein biochemistry of cancer cells.
So I think that's a good lead into your current work, because I mean, you've continued to be a leader in this field, still continuing to work on the mechanism by which cancers arise in cells. So you haven't spoken yet. What's the topic that you're going to be talking about at the symposium?
Well, by about the year 2000, 2001, I came to the conclusion, which of course was a bit premature, that we understood in outline how a normal cell becomes a cancer cell and forms a primary tumor. But we still didn't understand the last step of cancer development, which is how cells from the normal tumor, the primary tumor, travel away from the primary tumor and seed new cancer colonies in distant sites. That is the so-called process of metastasis. So starting in the 2001 or 2, people in my lab began to look for metastasis-inducing genes that empowered cancer cells from a primary tumor to travel to distant sites in the body. In 2004, indeed, there was one such gene discovered, which had previously been discovered in the context of early fruit fly development, where this gene is also involved, because the genes that convert a normal cell into a primary cancer cell, and ultimately a primary cancer cell into one that invades and metastasizes, those genes have been around, for better or worse, for 600 million years, which is about the distance between us and our distant cousins, the fruit flies. And so we soon came to learn that the way by which these metastasis genes worked was to take carcinoma cells, which are actually cells of an epithelial origin, in other words, an epithelial cell sheet of the type that covers our skin, and convert these epithelial cells of cancerous origin into cells that are more like connective tissue cells, which we call mesenchymal or mesenchymal, such as the connective tissue cells under our skin. And this process of going from an epithelial to a mesenchymal state is called the epithelial-mesenchymal transition, or the EMT. And tonight I will talk about the adventures my lab has had in exploring the ramifications of the EMT and what malignant characteristics it imparts to carcinoma cells.
So how many genes does one know how many genes are involved or needed, a minimal number that's needed for the EMT to occur in a cell?
Well, one can take a primary carcinoma cell and one can force it to go from the epithelial to mesenchymal state via the aforementioned EMT, and now one gets an aggressive cancer cell with even a single introduced EMT-inducing gene. And that cell, now converted from a more epithelial to a more mesenchymal state, is now empowered to disseminate to distant sites in the body. And that's, in one sense, very satisfying, because it means one can trace the ability to disseminate, to spread from one site to the other in the body, to a small number of master control genes. There aren't hundreds of ways to do this. It's a very small number of master control genes. That doesn't solve the whole problem, however. And it's the problem that one has not yet really attacked successfully. And it's as follows. Let's say we start out with a breast cancer cell in a primary tumor, and it disseminates, it spreads to the brain or the liver or the lungs or the bone marrow. We kind of understand how it does that, i.e. through the workings of the EMT program. The big challenge, however, is to understand how that disseminated cancer cell, which has now inserted itself in foreign distant tissues, how it makes a living once it gets there, because its site of destination, whether it be the brain or the bone marrow, is a very foreign terrain to which it's poorly adapted at the moment of its arrival. And therefore, how does it learn how to make a living there? We don't really understand that.
Is it known? No. It would be interesting to know how many potentially metastatic cells are circulating in the blood, and how many actually manage to establish themselves. Do you think that's a very small proportion?
It's a minute proportion. I can give you an example. Between 30 and 35% of women who are diagnosed with a primary breast cancer already have thousands of micro metastases, small clumps of metastatic cells, in their bone marrow on the day of diagnosis, which means the cancer has already spread cells. You might think that means that 30 or 35% of those of breast cancer patients will die from metastatic disease. But it's not true. Only about half of those women who initially come into the clinic with disseminated breast cancer cells actually will ever in their lifetimes develop metastatic breast cancer. And what that means is what you just implied, that the chance, fortunately, of a cancer cell that has wandered to a distant tissue of successfully founding a colony of breast cancer cells in the brain or the bone marrow is minute. Maybe 1 in 10,000, maybe 1 in a million. We don't really know the inefficiency of the process.
Do metastases from particular cancers like breast or liver, or do they have preferred sites of forming metastases? I mean, do metastases from breast arise more frequently, let's say the liver, than lung metastases? I mean, are different tissues equally good targets for all cancers?
And the answer is no. Some tissues have a predilection, a tendency to to generate metastases in certain sites. For example, prostate cancer typically creates metastases in the bone marrow. Colon cancer typically creates metastases in the liver. Breast cancer is less narrow, but it also creates bone marrow metastases and lung metastases and brain metastases. And I suspect, but I can't prove it to you, that the reason why there are these predilections, these tendencies, is that, for example, if a prostate cancer cell goes into the bone marrow, travels through the circulation into the bone marrow, it is readily able to adapt to the foreign microenvironment in which it's landed, whereas it has a very hard time figuring out how to make a living in the liver, for example. And therefore, even though the prostate cancer cells get spread all over the body, the metastases that arise are testimonials to the ease or difficulty of the cell of origin to make a living in one or another tissue.
So that also implies then that the microenvironment of the metastatic cell in the prostate presumably has some affinity with the microenvironment in the bone marrow, if that metastatic cell appears to be able to flourish there more readily than any other tissue.
I would agree with that, but articulate it slightly differently. It could be that a prostate cancer cell growing in the prostate, when it has to make fewer adaptations when it lands in the bone marrow than the number of adaptations or new behaviors it would need to acquire if it were to go to the brain. And of course, I use this word adaptations with some reluctance because we don't really know how cancer cells from one site of origin make a living in a distant site in the body.
Yeah, yeah. Would selection be a better word than adaptation? That the primary cell, the prostate cell, ends up in the bone marrow, and it struggles to survive, but may be able to do 2 or 3 divisions or something, and then you get selection by the microenvironment for progeny.
It's both in Darwinian terms. Let's say that a prostate cancer cell ends up in the bone marrow, and it may sit there for days, weeks, or years, trying with little success to make a living there. And one of its progeny may, through happenstance, acquire the skills, the adaptive program to enable that cell to start proliferating. So it's a model, albeit a crude one, of Darwinian evolution.
We must finish in just a moment, but let me finish with asking you, as I guess somebody who came off the street would ask you, does this work, these findings, have implications for dealing with metastases clinically?
I don't know. I could sell what I've just told you as being the answer to preventing metastases, and I wouldn't want to. My scientific trajectory has always been to try to figure out what's going on without being biased or influenced by necessarily how useful it's going to be in the clinic. Because to my mind, curiosity-driven research is much more likely to turn up unexpected insights that may in turn become useful to someone someday than is a highly directed and focused research program that is trying to, for example, develop a cure for metastatic disease.
Well, on that point, I think we'll finish. Thank you very much, Bob.
Glad to be here.